All right, the second major stage of protein synthesis. What is that? Translation. We have represented in this picture from your book all the key actors in translation. You first of all have purple blob. What's purple blob? In this book, it's our ribosome. We have yellow ribbon. What's yellow ribbon in this chapter anyway? Yellow ribbon is a messenger RNA molecule. So the ribosome is traveling down the messenger RNA molecule like a little train car down a track, chugga, 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 chugga. And what's the ribosome doing as it travels down the messenger RNA track? Reading the message. The message comes in little groups of 3 letters. What is each little group of 3 letters called? That's right, a codon. And so it so happens that ribosomes process 2 codons at a time, a total of 6 bases. 6 bases, 2 codons. And we see how this happens. It happens with the absolutely essential assistance of Mr. Blue Guy. What's Blue Guy? Blue Guy is a tRNA molecule, transfer RNA. Each transfer RNA brings 1 green block to the show. What do these green blocks represent in this book? They represent amino acids. And so amino acid is brought to the ribosome, and we see 3 bases at one end of the tRNA molecule that latch on to the messenger RNA. Ooh, 3 bases, codons. Yes, there are 3 bases from the tRNA molecule that latch on to 3 bases of the messenger RNA molecule. This side's a codon on the messenger RNA side. What's the tRNA side? It's called an anticodon. For every codon, there's a corresponding anticodon. And as the tRNA molecules bring base after base, they are hooked together in a growing amino acid chain. Well, let's look at this next picture and just a little more information about the tRNA molecule. Again, one end there is a green block or an amino acid. This end's called the amino acid attachment site. At the other end is what's called the messenger RNA attachment site. It is 3 bases. 3 bases called an anticodon. And so for every messenger RNA codon, there is a corresponding tRNA what? Anticodon. For every codon, there is a corresponding anticodon. So now you can tell me how many anticodons there are, can't you? Sure there are. Sure you can. The exact same number is codons. How many codons are there? Ooh, let's see. We got 4 letters taken 3 at a time. Let's see. What was that again? 4 times 4 times 4. 64. Yeah. There are 64 codons, 64 corresponding anticodons. So, what codon latches onto which anticodon? Well, here we've got RNA to RNA, and so it's simply A's to G's, A's to U's and G's to C's, A's to U's, G's to C's, and so we've got down here we've got CG, CG, and UA, A's to U's and G's to C's, RNA to RNA. So again, the tRNA molecule has a messenger RNA attachment site at one end, called the anticodon, and an amino acid attachment site at the other end. All right. Oh, here's the different types of tRNA in passing. We've already talked about 2, and ribosomes are also partly made of RNA, also some protein component, but RNA takes 3 forms, messenger RNA, tRNA, and ribosomal RNA. But back to translation. This is approximately the same picture we've had before, and so here's a tRNA molecule coming in for a landing, so to speak, and which tRNA molecule will come in for a landing? The one with the correct what? Anticodon. And so let's go to some pictures over in the next page. Take it from the top, and this picture emphasizes that ribosomes have actually 2 subunits, a small and a large. I will not ask you one thing about that in the test, but here we have the small subunit, and we have a tRNA molecule coming in for a landing. Which one? The one with the correct what? Anticodon. Is this correct? A's to U's, U's to A's, G's to C. Yep, absolutely. That tRNA molecule has landed. Going on to the next page, we have our next tRNA molecule coming in, again with the one with the correct anticodon. Is this correct? I see CG, UA, GC. Yep, right one, it's landed as well. And so now we have these tRNA molecules side by side and kind of holding hands there, it looks like. But now what happens next is crucial. There's some changes between this picture and this picture. Concentrate on the green blocks. What things have changed? Well, let's see. I look here. Oh my goodness. These two amino acids are now joined together. They weren't up here, but they are down here. Yeah, we know what that's called. It's called a peptide bond, right? An enzyme made that happen. We learned about that earlier. What else has changed? Well, it looks like that methionine, that is the full name, but it looks like this amino acid is no longer attached to the tRNA molecule. It's not. And so we had one attached, one unattached taking place, and then the ribosome moves down one codon. And what happens to the first amino acid, the one that used to have the met attached? It takes off. Another one comes in for a landing, and it repeats itself over and over again as the ribosome travels down the messenger RNA track. tRNA molecules come and go, come and go, come and go. Every time a tRNA molecule comes and goes, it leaves behind a what? An amino acid. So the amino acid chain gets longer and longer and longer until the ribosome does what? Reaches the end of the message on the messenger RNA molecule. So that, in a nutshell, is how translation works. All right, that's it for now.